Now coming to the requirements and technique used in RDNA technology. So coming to the tools or the requirements, I have mentioned few, but there are a number of requirement. So commonly these are called as tools of RDNA technology. The first one to name is enzymes. Again, enzymes, various enzymes are involved, but we will be dealing with two important enzymes, that is restriction endonucleases and the second one is ligases. The second important tool is the vector, which is nothing but a cloning vehicle. The third important tool is the host, which is nothing but a factory of cloning, where this is the place where cloning happens or amplification happens. Then the desired DNA or DNA to be cloned, this is one more tool or requirement. So coming to the techniques, already we have seen in the previous slide, there are so many techniques used in this RDNA technology for isolation, purification, identification, and few are gel electrophoresis, cloning DNA libraries, restriction enzyme mapping, polymerase chain reactions, nucleic acid hybridization, DNA microarrays, so on. So don't worry about all this, but you have to remember that these methods are used finally to have the expression of recombinant DNA. So now we will see one by one the tools required in RDNA technology. To start with, we will see the enzymes or the important enzymes involved in RDNA technology. As already mentioned, there are various enzymes which are required in this process. For example, restriction endonucleases, DNA ligases, polymerases, DNA polymerases. Already you have learnt in your replication, transcription and translation various enzymes. So all those enzymes are required in this process. But we will be dealing more about these two enzymes, that is restriction endonucleases, which cleaves DNA at a specific sites, and DNA ligases. They are the enzymes which join the two fragments of DNA end to end. Now we will see more about this restriction endonucleases. So restriction endonucleases, they are the molecular scissors which cut the desired DNA, and also they will cut the vector. So these restriction endonucleases are called as restriction enzymes because they restrict the viral replication in a bacteria, thereby preventing viral infection to the bacteria. That's why they are called as restriction enzymes. And one of the important points you have to remember is these restriction endonucleases are very specific in their action. I mean to say they cut the desired DNA at a specific sites. So they act on a specific sequence. So these specific sites or sequence are known as recognition sites or sequences. Recognition sites or sequence is the place where the desired DNA is cut by the restriction endonuclease. So how these recognition sites are? So these recognition sites, the sequence in these recognition sites are palindrome sequence. So what is this palindrome sequence? They are nothing but the inverted repeats. So if you read the sequence from 5 prime to 3 prime, they read same in both the strands. So we will look at this. It's a GAATTC. So this is the sequence in 5 prime to 3 prime in this strand, and you just observe the sequence in the below strand from 5 prime to 3 prime. So it is GAATTC. So both are reading the same when it is read from 5 prime to 3 prime. So such sequences are known as palindrome sequence. To understand it better, you can use the word Malayalam. So if you read from forward and backward, it reads the same. So such a type of sequences in DNA are recognized by restriction endonucleases, and that is the place where they act and cut the DNA. So when they act and cut the DNA, the cleavage pattern, you can have two types. The one is the sticky or a overlapping pattern, otherwise known as cohesive or staggered pattern. Another pattern which can be formed due to this action of restriction endonuclease is blunt end. So it can be a sticky end where there is complementary overlapping or overhangings, or non-overhangs or a uniform edges, that is the blunt ends. In this slide, you can better understand the cleavage pattern of restriction endonucleases. So now you can see the palindrome sequence. They read the same when it is read from 5' to 3' direction. So this is a double-stranded DNA segment. So now you can see this is known as palindrome sequence, which is a recognition site or sequence for the action of restriction endonuclease, or otherwise abbreviated as RE. So when an RE acts, so you can have two cleavage pattern. So one is a sticky and another one is a blunt end. So in sticky end, you can see there is a overhanging or a complementary basis. So this will actually helps in ligating the two DNA fragments very easily. So whenever you have a sticky ends, they are more preferred in rDNA technology than the blunt ends, where it's a uniform and there is no overhanging of the sequences. These are the few common examples for restriction endonucleases and the sources, that is from where they are being isolated, and also the type of cleavage they form when they cut the DNA. So, and you have to remember EcoR1 is the most commonly used restriction endonucleases. Now we will see the nomenclature of these restriction endonucleases. So they are named in a pattern. For example, we take this very important restriction endonuclease, that is EcoR1. So it is not RI, it is R1. So the first letter, which is written in capital, denotes the genus. So E stands for Escherichia. And this CO, which is in italics, actually denotes the species, that is coli. So it is Escherichia coli. Followed by we have one more letter, that is R. So this letter denotes the strain RY13. And finally we have a Roman numerical, which is an order of identification of this enzyme in that bacteria. So this is actually number, not a letter, which is a Roman numerical. So you should read it as EcoR1. Similarly, you can go back and see or name all those restriction endonucleases which we saw in the previous slide. So there are three types of restriction endonucleases: type 1, type 2 and type 3. Type 1, it cleaves DNA at random sites. It can be more than 1000 base pair from restriction sequence. And these will have endonuclease as well as methylase activity. And type 1, they require ATP for their action, that is they require energy for their activity. Coming to the type 2, they cleave DNA within the recognition sequence, and they are very simple and they don't require any ATP. So this feature of type 2 makes this type 2 as one of the important enzyme which is used in rDNA technology. Coming to type 3, so type 3 restriction endonucleases, they cleave DNA at about 25 base pair from the recognition sequence, and they also, like type 1, have endonuclease and methylase activity and they require ATP. So these are the types of restriction endonucleases, and for recombinant DNA technology, the preferred type is type 2 restriction endonucleases. We know that restriction endonucleases, they cut the DNA at specific sites, and these sites are called as recognition sites, which will have a palindromic sequence. And usually if you see the nucleotide base pairs, there will be somewhere around 4 to 7 nucleotide pairs. So when a segment of DNA from a species is made to react with a specific restriction endonucleases, a specific number of cut copies will be produced. So this is known as restriction map, and these fragments can be isolated by electrophoresis. So these specific number of fragments which is formed by the action of restriction endonuclease, they act as a fingerprint for that DNA molecule, because fragment of DNA from one organism will have a characteristic pattern. So it differs from one species to another species, or one organism to another organism. So we will understand with an example. So we will take a DNA fragment of lambda phage. Now we will have two endonucleases, EcoR1 and BAMH1. So we will see what happens if EcoR1 acts on this lambda phage DNA. So it cuts the DNA into these three fragments of these three variable lengths. So you can have a fragment of 21.8 base pair, 4.8 base pair and 5.5 base pair. So if EcoR1 acts, we have three fragments. Similarly, if the same lambda phage is made to react with BAMH1 endonucleases, restriction endonucleases, then we will be having 4 fragments. So these fragments are typical or specific for the DNA when a specific restriction endonuclease acts. So this cut pieces are specific number of cut pieces produced after the action of a restriction endonucleases is known as restriction map. Another enzyme which is involved in our DNA technology is ligases. So already during enzymology class, more about ligase has been told. So they are the enzymes which will join any two molecules. So ligases, here DNA ligases, they join the end-to-end ligation or end-to-end joining of DNA fragments. So the DNA can be one from the target DNA, other can be from the vector DNA. So they are the one which helps in joining these two fragments.